Good afternoon, everybody, and Ramadan Mubarak. I welcome you today for Ground Round, titled as The Use of Biologics and Biosimilar in Ophthalmology. I am Sita Zoman, I'm ambulatory care clinical pharmacist, and I will be the moderator for this session today. So while biologics and biosimilar have gained a significant traction in various medical fields, their adoption in the ophthalmology remains early. In fields such as rheumatology and oncology, biosimilar are already undergoing extensive clinical and integration into treatment protocols. However, in ophthalmology, there's a lot of concern regarding efficacy, safety, immunogenicity, and regulatory approval pathway. Continue to present a challenge leading to hesitation among clinician and researcher. This has resulted in what we can consider a gray zone, where more clarity is needed to guide clinical decision-making and policy development. Today, we are aimed for address this uncertainty by exploring the latest advancement in real-world application of biologics and biosimilar in ophthalmology. I am privileged to introduce our speaker today, Dr. Abdulmalik AlEissa, who is recently rejoined KKESH after completing his residency program in clinical pharmacy practice. He will provide a valuable insight into these therapies, current landscape, challenges, and future direction in ophthalmology care. I encourage you to participate in this discussion, asking questions, and contribute to this important conversation. So let's begin.
Assalamu alaikum. Good afternoon, ladies and gentlemen. I am Abdulmalik AlEissa from the pharmacy department. And before I begin, I would like to congratulate you the rest of Ramadan, وكل عام وانتم بخير, and happy Flag Day. Today we will discuss about one group of medication that has been changing the clinical practice in internal medicine, infectious disease, and ophthalmology, which are biologics and its copy biosimilars. Also in this lecture, we will track their development, also their approval by FDA and international agencies. First, I have no conflict of interest regarding this presentation. In this presentation, we will cover introduction to know what is the biologics and its history. And among the biologics, we choose the monoclonal antibodies and targeted molecules. Also, we will discuss the FDA regulation, biosimilars concept, and biosimilar in ophthalmology practice. So, biologics. If we look to the biologics as a word, it's derived from the bio, which is mean living. So the biologics are substances that made from the living organism or its products, and it's used in the prevention, diagnosis, and the treatment of cancer, infectious disease, and autoimmune disease. Biologics drug may include antibodies, interleukins, insulins, and vaccines. These agents or these drugs, if we compare it to the small molecules, we will find it as more complex and larger than small molecules. If we look to the pictures below, we can see the small molecules drug like aspirin, how it is smaller than monoclonal antibodies. So from the previous slide, we noticed the biologics, it's a broad term that may include vaccines, insulins, and monoclonal antibodies. From this timeline, we can see the first discovery for the biologics was in 1796 when Dr. Edward injected co-material of co-box onto an 8-year-old boy, then exposed him to the smallpox viruses, but disease not developed. This discovery was established the concept of the vaccination and immunology. In 1982, one of the pharmaceutical company developed the newer biologics, but that time was insulin. When they isolate a specific gene that's responsible to produce insulin and prepare the plasmid, which is considered as a carrier or a vector, they mix it together to keep these molecules as a recombinant DNA. Then they insert it to the one bacterium cell to produce a new generation of bacteria that's able to produce insulin. This discovery used now for treatment for diabetes. In 1986, US FDA approved mirumumab CD3 as the first monoclonal antibody, which used for the solid organ transplant rejection by hybridoma technology. We will discuss it later. So monoclonal antibody, it's type of antibody that's made by identical immune cell, which is B lymphocyte, which are all a clone of the single parent cell. This antibody monospecific, meaning they designed to target and bind to one specific antigen. They are used in the medical treatment for the cancer, autoimmune disease, and infections. One of the technology that used to produce monoclonal antibodies called hybridoma technology, when the Dr. George in 1975 challenged one of the mice with a specific antigen to stimulate their immune system to produce antibodies. This antibodies was isolated from B lymphocyte after spleen removal. Then they fuse it or mix it with myeloma cell. Myeloma cell, it is cancer cell, and the rationale behind this fusion, it is to keep this cell as an immortal, never-ending cells. Then they placed this hybridoma cell into one special medium called HAT medium to allow the fused cell to be survive. Then they screen it for harvest monoclonal antibodies. And we have some monoclonal antibodies, drugs that derived from that technology, for example, rituximab, infliximab, and muromonumab CD3. A newer technology was discovered by Dr. George Smith in 1985, but this technique was more complicated than the previous technology. He chose one of type of the virus called phage. He inserted a specific gene, mainly antibody fragment, into their DNA to allow them to be any growth in the phage surface, then display them or present them on the specific antigen or specific receptor, mainly drug-related antigens, to ensure the good antibody bindings. And he repeated 3 to 5 times to ensure the best antibodies bindings. After that, he elutes these antibodies from the phage. Then he regenerated or re-engineered it with full immunoglobulin to generate the Y shape of the antibodies. There is many monoclonal antibodies drug derived from that technology. For example, belimumab, ranibizumab, and adalimumab. There are several types of monoclonal antibody, depends on the source of the antibodies. For example, we have a murine, which is 100% derived from the mice. We have chimeric antibodies, which is part from the mice cells and the human cells. And we have humanized, which is a small part of the mouse and small part of the human cells. And we have fully humanized antibody. Also, we can humanize the monoclonal antibodies by biotechniques. It's basically by changing the binding site of the mice antibodies to the human antibodies. Targeted molecule, it is one of the biologics drugs. When we discovered any biologics, we have to keep these molecules as a targeted agent to understand the pathophysiology of some diseases. For example, we designed the targeted molecules that target that protein that play crucial role in the, for example, cancer, inflammation, and infectious disease. And if we take the aflibercept as an example, we will find that it will target VEGF-A and B and the placental growth factor, which play a crucial role in the blood vessel overgrowth and fluid leakage. Second example, we have faricimab in the same way. It will target VEGF-A and angiopoietin-2, which is also play a crucial role in the blood vessels overgrowth and fluid leakage. Adalimumab will target one of the pro-inflammatory cytokines to prevent them to interact with the TNF receptor 1 and TNF receptor 2. From this slide, let's agree the biologics agents, it is complex molecules and used for the rare disease. Based on that, international agency implemented some regulation to ensure their efficacy and their safety. If we look to this slide, as a researcher or the pharmaceutical company, if they want to develop any biologic, they should submit one form called investigational new drug application. This application should contain drug development process, preclinical research, which is mean pharmacokinetic, pharmacodynamic, and in vivo and in vitro studies. After that, once the application done by FDA, the researcher or the pharmaceutical company will be allowed to conduct 3 clinical trials. Phase 1, which is mainly focused in the safety and it's contain around less than 100 patients. Phase 2, mainly focused in the safety and contain 100 patients. Phase 3, it's contain 1,000 patients, larger population, and will ensure both efficacy and safety. Once the pharmaceutical company or the researcher finish from this trial, will apply a new drug application for FDA for the final approval. I would like to stop for a while for the phase 3 clinical trial because it's one of the final stage of a clinical trial that testing for a new drug before it's submitted for approval for the FDA. As I mentioned, phase 3 clinical trial, it's determine the overall effectiveness and safety of the drug. And also it contains larger population than phase 1 and phase 2. We can see most of the definitive clinical trials was phase 3. From this timeline, we can see the first, or not first, example for the biologics agent approval by FDA. In 1986, we have miromonomab, CD3, then rituximab. After that, we got infliximab, 1998. In 2002, adalimumab was introduced to the market. Bevacizumab was introduced in the market 2004. Ranizumab was approved in 2006. Tocilizumab was in 2010. 2020, we got teprotumumab for the thyroid eye disease. And 2022, we got faricimab. Biosimilar as a concept. There is international agencies defined the biosimilars as biologics medicine. It is considered a biologics medicine, highly similar to the another already approved by EMA, European Medicine Agency, and US FDA approved the biosimilar as a biologics also, highly similar to another biologic that approved already by FDA. Biosimilar as safe and effective as original products. Biosimilar may be costless because manufacturers rely on the FDA findings that the original biologics safe and effective. And if we say the timeline for the approval for the biosimilar compared to the biologic, we can see it's shorter than reference products or the biologic. Also, we can see the phase 2 was deleted from the biosimilar because it's copied molecules, it's copied drug. Also, we can see the required data to approve the biosimilar. If we look to the right side, the biosimilar, the basis was analytical assessment to ensure the similarity of the drug. While in the reference product or the original product, we can see the basis was a clinical study because it is novel products and a new product. Regarding Saudi FDA, biosimilar is defined as a therapeutic protein produced using recombinant DNA technology or gene expression method, designed to be highly similar to already licensed reference biotechnological product. It's similar to the international agency, EMA and US FDA. Also, there is a Saudi FDA guideline for the biosimilar to ensure their efficacy and safety. To date, FDA has approved 65 biosimilar, 14 of them as considered as interchangeable, and we will know what is the interchangeable meaning. Example for the biosimilar, we have adalimumab, aflibercept, infliximab, bevacizumab, and ranibizumab. Interchangeable product. Interchangeable is biosimilar, that means additional requirement and may be substituted for the reference product at the pharmacy depending on the state pharmacy law. Interchangeable biological product, also called interchangeable biosimilar or interchangeable product, may help increase the patient access to the biologics. One thing is important, not all biosimilar are interchangeable. Companies must submit the additional data to support their product to be interchangeable. For example, we have adalimumab, we have 10 of them as a biosimilar and 4 of them considered as interchangeable. Aflibercept, we have 2 biosimilar, both of them as interchangeable. ranibizumab, we have two biosimilar, and both of them are interchangeable. There is a statement from US FDA answer the most common question regarding the interchangeable product. I choose three of them. We will discuss it in the next slide. Question 1, can interchangeable product be substituted for the reference product by a pharmacist? The answer was yes, based on the last definition, but should be based on the pharmacy state law. Second question, what the approval standard do interchangeable products have to meet? The answer was required data, as I mentioned, and sufficient clinical studies. Question 3, should healthcare prescriber or provider be concerned if his or her patient received interchangeable product in place of the prescribed reference product? The answer, no. It's biosimilar, and it's show its efficacy and safety based on the clinical trials. Biosimilar in ophthalmic practice. If we look to the reference biologic that used in the ophthalmology practice, we can see ranibizumab. The first approval was 2006 for the NMD. 2011, we got aflibercept for the NMD. 2014, we got the adalimumab, the first biologic that approved for uveitis. 2020, we got teprotumumab for the thyroid eye disease. 2022, we get faricimab. This is considered as a reference biologics agents, not biosimilar. Biosimilar in ophthalmic practice, it's more similar to the FDA agreements or recommendation. And this recommendation was based on the American Academy of Ophthalmology. Biosimilar product has no clinically meaningful differences in terms of the safety, purity, and potency to the reference product and its labeled indications. So we should ensure that patient receive the biosimilar as a labeled indication. Biosimilar may be approved for all subset approved indication for the reference product. FDA does not study the safety potency of label used of biosimilar. And this is the timeline for the biosimilar that used in the ophthalmology practice. 2021, we got ranibizumab. 2023, we got ranibizumab as the first interchangeable product. 2024, we got 3 aflibercept as a biosimilar. The last one, the third aflibercept, is not approved yet. Summary of American Academy for the biosimilar recommendation. If biosimilar is suggested by a pharmacist, the ophthalmologist must review the FDA approved indication for the biosimilar. It's like US FDA recommendation. For off-label use, American Academy, again, substitution until strong clinical data support their efficacy and safety. So it is not absolute contraindication. Ophthalmologists should be aware about medical legal when they're using biosimilar, not FDA approved for the intravitreal use. Bevacizumab as a biosimilar have been developed for the cancer treatment, but they have not been studied for the ophthalmic use. And their inactive ingredient have not been approved for the eye. Academy strongly advise against using this biosimilar in step therapy or substitution for bevacizumab without sufficient clinical study, which is not absolute contraindication. This is the simple monograph for one of the biologics that commonly used in the ophthalmic practice, aflibercept. And this is one of the clinical trial. It's called INSIGHT trial, when they compare head-to-head study, the biosimilar versus the reference product. It's double-blind randomized clinical trial, multi-center clinical trial. The main outcome was primary outcomes was mean change in the best corrected visual acuity at week 8. If we look to the result at week 8, we can see the change in the best corrected visual acuity was almost 6 compared to the biosimilar, which is also almost 6, which is ensure their similarity. Also, the second outcomes was the safety. We can see both of them has similar results. 30 almost and 29. The conclusion of that study, the aflibercept biosimilar demonstrates the clinical equivalence in regard to the efficacy with comparable safety and to the reference aflibercept. Take-home message, biosimilar in ophthalmology are promising agent offering cost-effective alternative to the reference biologics with similar efficacy and safety to improve patient access to the treatment for eye disease. However, ongoing research efforts are needed to ensure the long-term safety and optimize their use in clinical practice. Thank you for attending this. Thank you.
Thank you, Dr. Abdulmalik, for this very informative lecture. And I have some question, or I'll give you only one question before we're starting with our audience. So comparing, according to you, that there is supposed to be a comparing study between the biosimilar and biologics. How do we compare the biosimilar to the reference biologics and regarding efficacy and safety?
The gold standard for any study, it is the randomized clinical trial, like this trial what I mentioned, the INSIGHT, and it is a challenge for the most of the biosimilars. We have limited number of clinical studies.
Comparing the
comparing as a head-to-head.
Okay, so regarding the cost, how does the cost affecting between the difference between the biosimilar and biologics will affect the patient access to a treatment?
Biosimilar as an agent, it's offering cost saving compared to the biologic or the original brand, which is also decrease the cost for the patient if he treated in the private hospital, also decrease the cost for the governmental hospital to receive this medication.
How do we expecting that they're going to be the difference? For how much is going to be usually the difference?
One of the study I found that they mentioned the costs will be from 15% to 50%.
Oh. Lower than the original price?
Lower than price.
Okay. Now I will give the audience the platform. Any questions?
I think it's a great to highlight what's about, biologics and biosimilar that nowadays, as we have a lot of biosimilar now starting to be appear in the market. And actually in our pack, finds that we have the original products and we get used to that. And now we are going to get, the biosimilar starting to be hesitant to use it or not to use it. So in regarding to intraocular, injection, especially for diabetic macular choroidal nephropathy, we are hesitating because we don't have enough, trial to support the biosimilar, although we have something, but I think it needs times. We don't like to compare things only in the cost, but because this is very sensitive issues. Regarding to systemic biologics and biosimilar, we have, starting to use it based on the rheumatologist, experience and efficacy. But we use it in our hospital, and Dr. Abdullah, he knows, and others. We have some sort of manufacturing of these medications, and let us to say the means that to give it as subcutaneous injection. There is an issues with it regarding to the allergy side effects. So do you have that concern? Because actually, as ophthalmologists, we are weak in this point, but actually our patient is really not really having good feedback about biosimilar when you compare it to the original.
Actually, we have to ensure, does the patient receive biosimilar as a labeled indication or not?
Of course. We have adalimumab and infliximab. Both of them were, if they approved for both, and then we have the biosimilar introduced last year in our hospital. The experience of our patient when we are shifting them actually wasn't that good, pleasant as compared, and this already.
It is recorded. Yes, there is some failed.
Regarding to effectiveness, some of them really is not effective as the original.
Maybe. Second question, does this medication consider as interchangeable agents? As I mentioned in the third table, we have 10 of infliximab as a biosimilar and 4 of them only as interchangeable. And for me, yes, it is one. Our product is one of the interchangeable products.
Product, and we get used to it.
I totally understand that usually because we are in the ophthalmic field that using biologics still, as I mentioned before, is a lot of hesitation regarding that. And yet, I think because adalimumab, the biosimilar has been already used for, and we use it here not only for the uveitis, we use it with another, usually uveitis come with us for patients who came in with us for uveitis with other disease like multiple sclerosis or other disease, rheumatology disease. So we actually, when we use it for treating the main cause, and uveitis usually is accompanying, we rarely receive a patient who has only uveitis when you have another rheumatology disease. I totally understand your concern, but I think that's what encouraging us to do more research regarding this biosimilar and those patients, considering that we are already a research center. So I think this is going to be one motivation for us to do more research biosimilar regarding uveitis patients. Yes. Other questions? Okay. Dr. Abdulmalik, do you see any future more indication for biologics or biosimilar use in ophthalmology, or you do not see anything? Is there any promising
In ophthalmology?
in ophthalmology? Yes.
Actually, for the biosimilar, there is recent INSIGHT clinical trial, the last trial that I mentioned. It will be, it was used to assess the efficacy and safety for the aflibercept to be approval this year, inshallah, 2025.
For the DME.
For the DME.
Oh, so this is good. This is a new promising
For
indication.
the other indications, one of the challenges for the infectious disease as practice is the antimicrobial resistance. Now we have two medications as a biologics using the HIV for the multi-drug resistant. It's called abalizumab, and C. diff, which is the intra-abdominal infection. We use tazocin sometimes, vancomycin, metronidazole, which lead to the recurrence, secondary to the resistant. Now we have new agents called bizalutuximab, which is a biological agent used for the C. diff. It will change and revolutionize the practice in the infectious disease.
That's very promising. Any other questions? I think now we conclude our session. Thank you for all the attendance, for attending our ground round. Hope it was informative. Thank you, Dr. Abdulmalik,
Thank you
for your presentation.
Thank you.
Thank you so much.